Welcome to the first episode ever of Science in Real Life. I'm Molly, and I'm an assistant research scientist in the Center for Genomics and Systems Biology at New York University. Today in the lab, I'll be extracting deoxyribonucleic acid, or as the cool kids call it, DNA, from plant tissue. And DNA is a really great place to start this series because it's the focus of so much science in real life. If you're a biologist like I am, chances are you're working with DNA or its derivatives, and as a student, you've probably spent a lot of time learning about DNA. Like, so much time. And you might be like, why do I have to learn about the history of this molecule, how it's replicated, transcribed, and translated, intimate details about its structure? I feel you, believe me. Here's the thing though. There's a better way to learn about DNA, and that's by doing science. Every single organism on this planet contains DNA. I contain DNA, you contain DNA, unless you're like a machine robot sent from the future to destroy us all, in which case skip this episode. DNA contains all of the information that makes you, you. It's organized in a way that lets us read its information like a book. The words in the book are our genes. The genes are responsible for our traits. And organisms pass on these genes to their offspring, and in this way we inherit dazzlingly blue eyes or debilitating genetic diseases. So in order to get the information that DNA contains, I have to isolate it first. And that's the DNA extraction experiment I'll be doing in the lab today. Okay, so when I do an experiment at my bench, I follow a series of steps called a protocol. It's a lot like a protocol that Jason Bourne or Jack Bauer would follow, except with less explosions, guns, and killing, and handsome men, but with so much more science. Now I have to go get the tissue that I'll be using for the DNA extraction, and it is very conveniently located in our beautiful growth chambers. We're using leaves from Arabidopsis thaliana plants. Arabidopsis is kind of like the guinea pig of the plant biology world. The DNA is hiding behind a lot of barriers in the cell. So first I need to get past the cell wall, which is made up of really strong cellulose. And I'm going to do that by grinding up the tissue in liquid nitrogen, which makes it look like a super legit science goddess. Liquid nitrogen is, well, nitrogen in its liquid state, which means it is so cold. When it comes into contact with the tissue, it flash freezes it and immediately preserves all of its components and makes it really brittle. So when I attack it with a mortar and pestle, the cell walls will break and I'm one step closer to getting the DNA. There are two barriers left. I have to get past the cell membrane, and then the DNA is still hidden away in the nucleus. If I add something that gets rid of membranes, then I can knock them both out in one step. So I'll add a buffer that contains detergent to my samples. Now that I've added the detergent, I'm going to let my samples stew to make sure those membranes are good and gone and my DNA is free. Now I'm going to add a buffer that gets rid of the detergent I added just now, and also removes any gross, large contaminating molecules. Now I'm just going to throw these in the centrifuge for a quick spin to pellet any gross cellular junk at the bottom of the tube. We've gotten rid of the big molecules, but the solution on top of the pellet still contains a bunch of other stuff from the cell, and we need to get the DNA out of that. To do this, I'm going to add a buffer that contains a high concentration of salt. Then I make the salty solution pass through a column made of silica by spinning it in a centrifuge. Under the high level of saltiness, the DNA will bind to the silica column, and everything else will flow into the tube, which I discard. Once my DNA is bound to the column, I really want to make sure that it gets extra super squeaky clean, so I'm going to wash the column with an ethanol solution. Now I need to get my clean DNA off the column and into the test tube. This step is called elution, and it's done when I add a buffer to the column that contains a lot less salt than before. This has the opposite effect of the really salty solution and makes the DNA lose its affinity for the silica. So when I centrifuge it, this time it's the DNA that will flow through into my tube. And now I have some beautiful Arabidopsis thaliana DNA in my test tube, and I can measure how much I have using our nanospectrophotometer. I load just a teeny bit of my sample onto the nanodrop, and it'll tell me how much DNA is in my whole sample by passing light through it. Woohoo! We have 99.5 nanograms of DNA per microliter of sample, so my DNA extraction worked. The DNA in these tubes contains the organism's entire genome. It's like an amazing instruction manual for how to be Arabidopsis. And I can see if this DNA contains genes that would help the plant survive a drought or use fertilizer more efficiently, or to realize my dream of making an Arabidopsis Jurassic Park. Rawr!